Hello, my name is Seema Ibiebula, and this is my report on the blood test, a study of myoglobin conformation. First, let's give some background on what myoglobin is. Myoglobin is a protein that is found in your striated muscles. This includes your skeletal muscles and your heart muscles. Its main function is to supply oxygen to the cells in your muscles. The image on the right is of a myoglobin protein. The green corresponds with the heme group, and the red corresponds with molecular oxygen. The blue is the rest of the protein. Next, let's talk about denaturation. Protein denaturation is when the tertiary structure of a protein is lost. This is otherwise known as protein unfolding. Protein denaturation can be brought on by physical or chemical agents. Examples of physical agents would be heat or radiation, and examples of chemical agents could be acids, alkalines, etc. In this experiment, we tested the absorbance of myoglobin solutions with different concentrations of denaturant. The denaturant we used is guanidine hydrochloride, which can be seen to the right. Guanidine is a natural product of protein metabolism that could be found in our bodies. The goal of this experiment is to determine the equilibrium constant for the denaturation process of myoglobin, the Gibbs free energy associated with this process, the rate of denaturation, and the concentration of guanidine hydrochloride where the amount of naive and unfolded forms of myoglobin are equal. Since myoglobin is such an essential protein in our body, it is important to understand the process of its denaturation. Before doing the experiment, I came up with two hypotheses. Hypothesis number one is that since myoglobin has a very stable structure at most concentrations of guanidine hydrochloride, it will continue to be in the native state. Hypothesis number two is that samples 17, 18, and 19 will have the unfolded form of myoglobin due to the high concentrations of guanidine hydrochloride. The experimental procedure for this lab was fairly simple. We made 19 different samples. Each sample was 5 milliliters total and was made up of myoglobin solution, buffer, and guanidine hydrochloride. They all had the same amount of myoglobin solution at 1 milliliter. They had different amounts of guanidine hydrochloride and buffer solution, so each sample would have a different concentration of guanidine hydrochloride, with the concentrations ranging from 0 to 2.4 molar. We then ran each sample through a UV-vis spectrophotometer to get absorbance values. In the graph below, you can see the absorption spectra for all 19 runs. The data is also listed in the table next to the graph. There are a few trends of note that can be seen in this data. The first trend of note is that as the concentration of guanidine hydrochloride increased, the absorbance of the samples decreased. This is due to the decreasing amount of the native myoglobin being present in the samples. The other trend of note is the wavelength of the Saurit band. The Saurit band is the intense peak in the blue wavelength region of the visible spectrum, and it is our max peak here. As you can see on the table, in general, as the concentration of denaturant increased, the wavelength of the maximum absorbance decreased. Next, I made a plot of absorbance versus the concentration of guanidine hydrochloride. The portions of the graph outside of the outline box shows that in excess amounts and negligible amounts of denaturant, the amount of native versus unfolded myoglobin doesn't change much. From this graph, you can tell at concentrations below 0.9 molar, myoglobin is almost completely folded in the body. The concentrations above 1.5 molar myoglobin is almost entirely unfolded in the body. The values on the side next to the graph of AU and AN were taken from the endpoints. AU is the absorbance in extreme excess of denaturant. For my AU value, I use the last mark on the plot. AN is the absorbance in complete absence of denaturant. I use the initial mark on the plot for this value. As one of my hypotheses, I stated that samples 17, 18, and 19 will have the unfolded form of myoglobin due to the high concentrations of guanidine hydrochloride. And in this plot, samples 17, 18, 19 are in the endpoints with absorbance values consistent with AU. This supports my hypothesis. The outline part of the graph is where the absorbance and concentration of denaturant correspond linearly. This is the concentration of guanidine hydrochloride where the denaturation process becomes significant. This part of the graph was used for further analysis. This graph also disproves my first hypothesis that myoglobin will be in the native states at most concentrations of guanidine hydrochloride, because it was only consistently in the native state up to concentrations of 0.9 molar. Next, I calculated the equilibrium constant, the Gibbs free energy value of unfolding, which is delta GU, the rate of denaturation, the free energy of unfolding in pure water, and the concentration of guanidine hydrochloride where the native myoglobin concentration is equal to the unfolded myoglobin concentration. The equilibrium constant was calculated for each sample in Excel using this equation. The Gibbs free energy of unfolding was calculated in Excel using this equation and the K values we just derived. This plot shows the K and delta G values for the concentration of guanidine hydrochloride that corresponded linearly with absorbance. I then used the delta G values and the concentration of denaturant to make this graph. The line of best fit of this graph corresponds with this delta GU equation. The M value is equal to the slope of the graph. So it is equal to 34,204. This value is quite large and indicates that once denaturation begins, it happens very rapidly. The delta G of water corresponds with the y-intercept of this graph, which is equal to 35,373. The CM value was found by setting y equal to 0 and solving for x, and this value is equal to 1.03 molar. The experimental value of the fraction of unfolded proteins was found in Excel using the first equation listed here. The theoretical value of the fraction of the unfolded protein was found using the second equation listed on the slide, as well as the solver added in Excel to improve the theoretical values of M and CM by minimizing chi-squared. The graph on the right is a plot of both the experimental and theoretical values for F versus the concentration of guanidine hydrochloride. The values for F experimental and F theoretical matched up very well here. To wrap up, myoglobin is an essential protein in the human body that helps oxygenate our muscles. Guanidine is a denaturant that appears in our body as a natural product of protein metabolism. From this experiment, we know that a concentration of guanidine hydrochloride of 0.9 molar, the denaturation of myoglobin becomes significant, and by a concentration of 1.5 molar, almost all the myoglobin in the body is unfolded. This experiment is just the first step in analyzing the denaturation of myoglobin. There are other agents that can also affect myoglobin. Future experiments could look into the effect of other natural denaturants or even temperature on the structure of myoglobin. Thank you.